very much to the organizers for having me here today. It's a pleasure to be here. So today I'm going to talk about, so I'm a multidisciplinary scientist with an interest in application research, particularly in regenerative medicine. And so today I'm going to talk about how 3D printing has influenced my work in tissue regeneration, tissue engineering work, and regenerative medicine. So this work is really, I have aimed to, with the intention of improving healthcare, and also as an educator, I have used 3D printing to teach students so that the next generation are ready as early adopters. So my talk will be based on this. I will briefly introduce some of the general 3D printing applications in regenerative medicine, and by no means I can't do justice in such a short time for the amazing work that's being done around the world, but I'll just mention that as an introduction to the work that I'm doing in developing elastic tissue with the intention of making it as a point of care system, and also finally just look at some of the regenerative medicine studies, how they can be addressed with 3D printing to have more accelerated outcomes. So I think all of us we are acutely aware of the need for artificial tissue and organs, because not just now, from a really long time ago, this has been an interest for a lot of people. And as you can see from this painting, it's been recognized even in that conversation of the need or the intention to regenerate or transplant limbs. And it has evolved very significantly. And early in the 20th century, this is Alexis Carrel, who's I think the father of vascular surgery, who introduced anastomosis. And he looked at how we can preserve organs with perfusion outside the body, and again with the interest of how we can really preserve organs and with the intention for transplanting. So regenerative medicine really, and tissue engineering, it's a very broad term, and it's used very loosely. It's really a play between cells, biomaterials, and vascularization to induce homeostasis in a diseased or damaged system. So that's really what we are trying to do, to introduce homeostasis in a system that's diseased and damaged. And for this, in the talk, I will be talking about how biomaterials are processed, particularly elastic material, with biofunctionalization and for disease-specific and device-specific purposes. So I looked at 3D printing relatively recently and looked at all of the applications of this as would be applied in surgery, because I worked in a department of surgery and with students. And we looked at how it can be applied not only for repairing and regenerating tissue, but also as a mode of communication with patients. Can we have models to explain disease conditions to patients and then therefore to ease their problems, and how it can be used as surgical guides perhaps, and also, and obviously prosthesis and other implants, etc. So this is an article, it's written to a very general audience. And so obviously there's, in addition to tissue regenerative implants, then there's prosthetics, which is well known. And that's one of the areas that's very well developed. 3D printing is used for mass production as well as bespoke implants, bespoke prostheses. And that's almost a very, it's very well developed at the moment. And looking at the mid-phase, there's an interest in using 3D printing to address problems with injury to the bone near the eye, which is very important at the moment because there's off-the-shelf available implants that are used, based on titanium, but if you can really make it to be patient-specific with 3D printing, it can have a significant outcome. So, so I was told by orbital surgeons and max-fac surgeons. And then there's this interest in making stents as implants that actually is staying in the body. And predominantly, so far, there's PLA has been used for as splints for tracheal regeneration, repair, but they're merely stents, not for regeneration, but still an application. So 3D printing has influenced the repairing of these structures. And then there's this complete tissue engineering that 3D printing is used for with bioprinters. And the principle here is that you collect cells from a person and you expand it and then place layer by layer with 3D printing technology. And then you mature the tissue to have the structure that you want. And this is very clever. However, I worry if that's very, how rapidly that solution can be available to patients, because as you would know, you have to collect cells and then grow them to have enough so that they can be used for to making an implant, and then again grow them again for a period of time. And then also this cannot be done anywhere, everywhere. It has to be done within a very specific labs. So it's for very special people that this technology can be available. It's very clever though. But my interest is in how we can use 3D printing to make, to have solutions, tissue replacement solutions, with minimal laboratory use, and also not having to rely on patient cells so much. And again, well, they were used as, this is used as models, but relatively recently it's been shown to have been implanted in a monkey using autologous adipose tissue, which is very impressive. So as I mentioned, my interest is in making tissue with minimal dependency on patient cells or having specific labs, so that it can be used to address concerns around the world where if you don't have these special conditions or the patient cell population is not available, and also if it's an emergency case, so if it's not elective, you would have to have a rapid solution. So how can we use, what solutions can we have to address these things? And 3D printing has shown is showing that this can be addressed in this way. So I'm interested in, to do this, I've been looking at elastomers, thermoplastic, which is a material that can be molded and remolded after heating, as opposed to thermoset, which is permanently there. I've been collaborating with the company Bioactive. They have been producing, they're experts in polymer, and they have been giving us this material to work with. And I have initially used the thermoplastic material to create structures in conventional methods. So to do this, I looked at what sort of platform can I establish to test this material. And I chose to look at tubular structures. So if you think your body has got a lot of tubular structures, and they could be in different sizes, but most of them are tubular. So once you establish a system that you can use a material to address tubular structures, they can be then streamlined to address many other conditions. So I looked at initially conventional methods of manipulating thermoplastic polyurethane, i.e. you might be familiar with casting, coagulating, electrospinning, electrospraying. So I used almost every technique possible to establish tubular structures. However, there are still things that can't be addressed with these conventional methods. So then I looked at 3D printing because it's actually allowing It's quite enabling technique that it allows concept to execution a lot more rapidly than conventional methods, not in most cases, in the case of developing elastic tissue. So, as I mentioned, I looked at a lot of tubular structures, and with 3D printing, I was able to produce a multitude of structures with a simple change, whereas in a code, whereas with conventional methods, I have to make molds and a lot of massive changes to be able to produce one little change in a structure. So 3D printing allows me to make a lot of differences with a range of mechanical properties in a short period of time, and then to be able to test if that's okay and if it's really mimicking biological conditions. And to take this further and to really establish a prototype, I decided to look at trachea, which is another tubular structure, but it is a relatively more sophisticated in the sense that it has this softer trachealis muscle at the back and also these cartilage rings around it. And so 3D printing was, if I were to move forwards with this technique, it's an excellent model to test structural and mechanical reproducibility of the technique and also the material compatibility. So this is actually, we've taken scans of patients and we decided to test its ability to replicate. But this is actually a student exercise that, as I mentioned, I was interested in getting students to do this work. And these are medical students, undergraduate students, and surprisingly, even we talk about 3D printing so much, but tomorrow's surgeons, some of them are, it's very special for someone to be using 3D printing, but that shouldn't be the case. It should be very common, and it should be just like histology, biochemistry. I think that's how it should be. But so I was very keen for the students to learn this, and I introduced this 3D printing in their modules. This is actually a replication of the structure, not a mimic, as an exercise to understand this. But this is what we really wanted to do. We wanted to be able to mimic the tracheal structure so that it behaves like behaves in the biologically, biomechanics are similar to a natural structure. So we use the two types of TPU, which is, one is a harder structure, which is TPU 90, to mimic the cartilage rings. And then a softer, relatively softer TPU to mimic the trachealis muscle and the intermediate tissues. We used FDM technique and created these structures. And these are printed to be adult-sized tracheas. And then we wanted to see if it is, we were successful in creating a structurally similar trachea, but we wanted to see it's absolutely behaving like a real trachea. So we wanted to subject the structures to the mechanical forces that a real trachea would be subjected to. And if you know, you would know that the trachea is longitudinally flexible, so you can move your head, and, but it has to be radially rigid. So we applied mechanical forces to the places where it should bend, and it bends accordingly, because one of the problems with other methods is that maybe it doesn't bend, but it sort of makes it's not flexible, but it just, how shall I say, bends in half. So it shouldn't create it should move without having a closure of the lumen. So that's what we wanted to create. And then we wanted to see if it's okay when it's attached to the rest of the respiratory tract. We obtained a porcine a lung system, and this is a video. I wanted to play that. To be able to see that it actually facilitates the airflow, and expands and constricts the lungs accordingly, and which it did. And we also tested its compatibility when it's attached to a suture, that it would be okay. So we were successful in clarifying this. The other thing is that trachea, as I mentioned, radially it should be rigid, but at the same time it should facilitate natural forces such as coughing, crying, and it shouldn't collapse. And this is another problem with the alternative available structures at the moment. They are either they're too rigid or they collapse. So we managed to find a medium between. So the very bottom one is a porcine trachea, and as you can see, it doesn't have this trachealis muscle as for a human, but we used it as a reference point to see the rigidity. And our tracheas, and we placed a Doppler scan to see these flexes, and we introduced very high pressure to the system so that it to observe its behavior. The very top one is a longitudinal view, and the middle one is a cross view. It moves okay, but there could be a little bit more flexibility, but this is what we achieved, and this is a great achievement with the flexibility. Not as close as the porcine trachea yet. So that's the structural and mechanical properties we've managed to achieve with 3D printing, much more closer to a real trachea. And zooming in, we wanted the structure to be the surface that the biological components would interact with to be similar to the natural tissue. So with 3D printing, we are able to create a multitude of structures in the microscale so that the cells can behave more favorably when in biological conditions. And this is not very possible with conventional methods. If so, these small changes were done very rapidly within a few hours. If we were to do these things conventionally, it would take days. We would have to make casts and molds and then cast them, and still it might not be very very reproducible as we have here. And so not only to see the interaction with cells, these structures, these sort of structures are important for us to be able to understand the cell behavior, because as you can see here, the cells really feel their environment and they respond to the environment. And this is important because tomorrow if you were to design a structure and if you want a particular cell type to respond in a certain way, understanding the cell behavior on a given structure is very important so that we can create a structure where we can predict the outcome. And we've observed this time and time again, a really small change can really have a significant difference in the cell behavior. We've observed that we've studied with human dermal fibroblasts and bronchial epithelial cells, and they behave significantly differently to different densities, different patterns, but I haven't yet studied why they're behaving, but I know that they're behaving, so that's another study if someone wants to do that. So these are some, like I mentioned, the cells are intelligent. If you provide them with the right environment, they can respond to these different changes. The microstructures changes, they're very sensitive to. So in addition to changing the topography, so to say, we can also change the chemical environment by introducing additives. So I've been looking at the influence of silver nanoparticles, for example, and nitric oxide donors. They're very useful as antibacterial agents. And also that silver nanoparticles and nitric oxide also has an influence in it is known for antibacterial properties, and also it is, we looked at nitric oxide as for easing breathing difficulties in children. So with the intention of introducing these particles into the respiratory tract stents, so that as an alternative to having the children attached to ventilators, can we have stents that deliver nitric oxide and then really make a difference? So how do we introduce these bioactive materials to 3D printed structures? We didn't want to, at this stage, we didn't want to, we wanted the bioactive molecules to be on the surface. So we looked at a pre-polymer. Pre-polymer is a polymer that's not gone to completion and that has reactive groups that could then interact with the bioactive molecules of interest. So we use that as almost as the glue that could interact with the bioactive molecules and the printed material, and then introduce these active molecules to the printed structures so that it has the bioactive molecules. And also, I came across this group that works with carbon nanotubes some time back, a very interesting group that's, they're doing a lot of manipulations with nanocarbon nanotubes. So I wanted to introduce nitric oxide donors into these tubes as a delivery system so that we can control the delivery of nitric oxide for the purposes that I mentioned in a more controlled and a prolonged way. That is one of the problems at the moment. You can have nitric oxide donors, but when you try to introduce that within an implant, it releases so fast, and that's not very good. So we wanted a system that can control the release and have it for a prolonged period. So with this intention, I started working with this group, and then I got interested in composites that can be electrical conductive. But relatively recently, I found that that they have already used TPU and incorporated graphene as for this purpose. So that's very interesting to know that's happening. So with interest in electrical conductive material, I got interested in its effect in healing, wound healing, and also for regenerative applications. And we looked at how we can introduce this conductive material or electrical therapy, for example, and to increase healing. And this is an example of how we've looked at measuring an area of wound, and therefore we can really tailor our delivery of the bioactive components. However, this is a model that we've done, and we still have to work on the delivery system. So the idea is that we can really mimic the different layers of the wound, and therefore have a more bespoke solution. And also this could be interesting for pressure therapy, negative pressure, so you can concentrate on a given area more specific to the person. And I came across another group that has done looked at wound patches with nanocellulose. Apparently Finland has a lot of nanocellulose, and they work with these patches. And it's interesting to see that nanocellulose is used for this because it absorbs a lot of moist that there's a wound exudates. And what will be interesting with 3D printing is if we can really have a system where if we can reduce the number of times the wound dressing has to be done, maybe as a more, if we can have it evaporating a little bit more, or have, so they're using electrical conductivity here together with this nanocellulose. That is something that's interesting. So with the interest of electrical conductivity, I was interested in how 3D printing was used for conductive systems. And as you know, neurosensory pathways are quite interesting, and there's a huge interest in how we can look at implants and really regenerate, to help regenerate or repair problems. And one of the examples is retinal implants, where if your photoreceptors are damaged, you can introduce electrodes to send signals and enable people to see that is not when it's not possible. However, the clarity of this vision depends on the number of electrodes. So to start with, for example, you might have come across the system Argus 2 or nanoretina maybe. The clarity really, if you start with, they started with 4 to 9 electrodes, it was almost like a blur, and then afterwards, gradually the vision improved with the number of electrodes. So you can imagine the limitation was the number of electrodes, and 3D printing really can have an influence here by introducing this type of electrodes and have a solution where it's not possible in conventional methods. And these are implants that's done quite widely, and 3D printing is doing a great job. However, another example is that in hearing loss, where the hair cells are lost, this is how the hair cells look like. And at the moment, you might know, once you lose your hair cells in the inner ear, they don't regenerate. So it happens with old age, or if you listen to loud music, toxins and things like that, could damage your hair cells in your inner ear, and they don't regenerate. And at the moment, there's a lot of research interest with stem cells, trying to regenerate this, in addition to having these cochlear implants. So at the bottom, on the top, you can see that 3D printing has produced these sort of ciliary structures. And this is another area that can be used, 3D printing can be used to really accelerate the repair of a structure where the conventional methods might be a bit far away. And the other example with the cilia structure is the trachea. I mentioned to you before, we managed to have a structurally and mechanically compatible biomimetic structure as an adult, but what we didn't address there was the structure of the epithelia, which is the biological inner component. Now, it's not enough to have just cells inside the structure, but they have to be functionally active. So what's the role of epithelium, which is the inner layer of the trachea? That's to prevent, it should prevent from bacterial infections. So for that, we propose to use silver nanoparticles, and therefore we don't rely on the cells. And if you can really apply, if someone can really work on this cilial structure for the inner layer of trachea, I think that will be marvelous. So it will be more rapid than going through the whole pathway of generating cells and this whole cellular regeneration. It has a lot of unpredictability, whereas if you go with this, you really can have a biomimetic structure, and that could be really point of care, so you don't have to rely on extensive cell culture, etc. And this is another very important area that 3D printing can be used. Vascularization, you can have the best structure that you want, but if it's not vascularized enough, it might not be very compatible, and the body might not be integrated well. So it's really important that that's addressed, and bioinks could be one of the areas that could be used to address this. So I hope I mentioned a little bit about how 3D printing can be used and how it can be used to either to accelerate the regenerative medicine solutions, or how it's really used to make prototypes that is difficult in other methods, like conventional methods. So, and hope that inspired you, and if you're doing research and if you would like to do these things, that would be really wonderful.